So welcome everyone. Thank you very much for joining this last webinar in a series of 4 organized by RealiseD on enhancing patient centricity in rare disease clinical trials. And now we'll have to warn you that this session will be recorded. So if you do not want to be visible on the camera, now is the time to switch off your camera, perhaps. So this session will be recorded and will also be made available afterwards for you to listen to again if you wish. As I said, this is the last webinar of a series of 4, and all 4 of them will be made available later on for you to listen to. We've had some very interesting discussions in the previous webinars, and we hope to continue that, of course, today. But I'm sure that will surely happen with this webinar as well, because we have an interesting set of speakers and panelists for you ready today. There is also from RealiseD a survey that has been launched that seeks to identify and understand some factors that influence the enrollment in clinical trials, specifically in rare diseases. So we invite you all to scan these QR codes that will lead you to this survey. So the more answers that we have from people involved in running clinical trials in rare diseases, the better, of course, our information will be on setting steps forward in improving the enrollment of rare disease clinical trials. So please go ahead and scan these QR codes. I would not recommend you to complete it now while listening to the webinar, but you can do that after the webinar, of course. Some housekeeping rules. So for everyone, to keep this as smooth as possible, we ask you to mute your microphones if you're not speaking. Of course, if you want to speak, you can raise your hand. We will have a Q&A session at the end of the session open to everyone, so you can raise your hand and then unmute yourself when we give you the floor. If you want to ask a question in the meantime, there is also a Q&A function, and that should be visible on your screen. And you can at any time submit a question there, and then the moderator Geert will then ask your question in between or at the end of these sessions. So feel free to make use of this Q&A function. And then we have the introduction. I'm not sure if Geert is already present in the meantime. I'm not hearing anything, so I will just continue. But this is the agenda for you for now. So we have Professor Geert Molenberghs will be the moderator. Hopefully, he can connect. Professor at statistics professor at the University of Hasselt and KU Leuven, and also the academic lead, project lead in the work package 5. We have a presentation of Rudradev Sengupta, who is a senior trial design lead at 123 company in Belgium. And then we have an interesting list of panelists for you to give their view on the topic of today. And we will end with a Q&A open for everyone to ask their questions. So without further ado, Rudra, I will stop sharing my screen and then give you the floor to share your slide. And we are very looking forward to your presentation.
Perfect. Do you see my screen?
Yes, we can.
Okay, perfect. Thanks, Johan, for the introduction, and thanks for the invitation for this webinar. So I will try to discuss, like you mentioned, that how we can focus more on patient centricity in rare disease trials. First, I I picked actually 3 questions that that the audience were asked during the registration, and I noticed that, well, most of you consider the biggest barrier is for a new trial design or adopting a innovative trial design is the concerns from the authorities. But also there is a lack of education or familiarity, and which brings me to my next question is how you were asked how familiar you were with multi-component outcomes or hierarchical endpoints. And it seems like for many of you, like 30% of you, it's very new. But some of you have already used it or at least aware about multi-component outcomes. So, and to the last question is basically what everyone wants to gain from this webinar. Uh, it's about the general understanding of the method or tools that you can use. So or practical examples. So I will try to make sure that everyone can take something back from this webinar. At least that's what I try to make my slides based on that as well. Okay. So traditionally, clinical trials, we know that it's based on single outcomes, right? So mostly focused on efficacy. But what we want to discuss today is more about how we can look at multi-component outcome measures, because it's not only efficacy that matters, but it's also safety outcomes. You can think about quality of life as well. So it's a multifaceted value of the treatment that we want to capture. Okay. Now, this brings me back also to the first question that when we want to incorporate a new methodology in design, there is regulatory concern, there is lack of familiarity. So we want to focus first on definitely the theory part, like it should be built when you're proposing a new design method, it should be definitely built on a strong theory, right? And I promise not to bore everyone from by statistical equations in all the slides, but we need to understand the basics of the theory, right? So it all starts from Wilcoxon-Ranksome's test. And what it says is if you have two groups, let's say treatment and control, uh, you order all the elements of the two groups, you give them ranks, and then you sum the ranks of the elements of X, right? That's how you define the test statistics. It has a known distribution, and you can do your hypothesis testing. Now, what is definitely now if you have this high value of the use of the statistics, that means you have higher ranks for X, and then that's basically what you want to test, right? And sometimes what is this notion of ranking? Ranking is essentially what you do by pairwise comparison. So you want to check like, okay, this value in X, so this value in treatment is higher than this value in control. So, or higher than those many values in the control group. So that's how you associate the ranks. And that's how the Man-Whitney late in 1947 showed that the idea of Wilcoxon-Rank-Sum test is essentially similar to the pairwise comparisons. And we want to generalize this idea by moving from pairwise comparisons to generalized pairwise comparisons. And how we do that is just not by looking at the values which are greater or less or equal to, but we want to do it by defining better or worse, right? So it seems a simple transition, but what is very important here to understand that this better or worse can be dependent on the context. And this way you can also integrate different kinds of outcomes in the same approach, which is the big leap. And what we do using this generalized pairwise comparison is you compare each patient in one group against each patient in another group, and then you give it a score of 1, 0, or -1, right? So if a comparison is favorable, you give it a score of 1. If a comparison is unfavorable to the treatment, you give it a score of -1. If it's a neutral pair, then you give it a score of 0. And then you sum these scores, which essentially gives you the number of favorable and the number of unfavorable pairs. And we define a simple metric called net treatment benefit, simply by which is the difference between the proportion of favorable pairs and the proportion of unfavorable pairs. Okay. So in essence, net treatment benefit gives you the difference between two probabilities. And it's essentially the net probability of doing better on the treatment rather than on the control. And this idea can be generalized in terms of multiple prioritized outcomes, different kinds of outcomes. It can be binary, time to event, continuous, all in a singular approach. So that's, let's say, that's the theory part. Maybe it seems very abstract for the moment. So we will look at a case study to better understand how this can help us to analyze randomized clinical trials. Now, this is a example of a pediatric epilepsy trial. And as some of you might be aware, that normally in this kind of trials, seizures are the most important outcomes, right? So we want to focus on the number of epilepsy episodes. But what about other outcomes like seizure duration, motor skill, communication skills? So there are other important outcomes in this context, right? So in this approach, or within the framework of net treatment benefit, we want to integrate these different kind of outcomes. And how we do this? So first, you look at the number of epilepsy episodes. You look at all possible pairs among the two groups. And let's say there are 10 patients in the experimental, there are 10 patients in the control. So you have 100 pairs in total, okay? 100 pairwise comparisons. And for this, what you do is you check, okay, for a particular pair, if this favors the treatment or if this favors the control. For if you just focus, this is an example based on simulated data, and if we just focus on epilepsy episodes, we see, okay, 34% of the pairs favors the treatment, 22% favors the control. So essentially what we saw, the methodology, the net treatment benefit will be 34 minus 22, that's 12%. But there is still 44% of the pairs which basically does not contribute to this single outcome-based analysis. And we want to use this 44% of the pairs to draw conclusions based on other outcomes. Next one will be seizure duration. So for seizure duration, we again look at how many percent of the pairs is better in favor of the treatment, how many in favor of the control, and we look at the net treatment benefit corresponding to that. Okay. The same goes for the motor skill, for the communication skills, and if you do a sum over the last columns of the net treatment benefit, you get a 21% of cumulative net treatment benefit. The beauty of this method is that you also see the individual contributions of each outcome, right? So if we had just focused on epilepsy episodes, we would have just gotten 12% of NTB, whereas by focusing on the others, you gain this and you reach up to 21% of NTB. So essentially, NTB uses this more information from the same amount of data and tries to de-risk your trial, okay? And if you can think of in the context of the rare disease also, the number needed to treat is just the reciprocal of net treatment benefit. So with 12% of net treatment benefit, you will have like 8 number needed to treat, which means like on average, you need to treat 8 patients to see a treatment effect. Whereas with 21%, it's basically you need to treat on average 5 patients basically. So it intuitively, because it tries to capture more information out of the same data, it helps you to gain the statistical power or reduce the sample size in different cases. Also, it's already more more focus on the outcomes that can matter to the patients more, just not the seizure episodes, but also you can think there is ways to incorporate clinically relevant thresholds here, um, to make it more patient-centric. So for example, you can think of that, okay, there is a gain in epilepsy episodes, but maybe that is not enough. And I will come back to it at a later stage when I demonstrate another tool. Hopefully that will make it more clear to you, okay? So now we see that, okay, there is 21% of net treatment benefit. And how do we interpret it? Like I said, that in terms of the number needed to treat, and it's also related in a way to the hazard ratio if you consider the single outcome. So in our experience, when you have more than 20% net treatment benefit, you can interpret it as a very effective treatment, and accordingly, okay? So now that we know how we can analyze trials using NTBs, but also we want to integrate this as in the trial design, right? In the same context, like I said, that normally it's seizure is the most important outcome in this particular setting, right? But FDA has a different guideline. So it suggests that you look at the change in mean seizure frequency, but that is affected by extreme values. EMA has a different way of looking at it. So, okay, if the reduction in seizure frequency is more than at least 50%, then it's a responder. So you do responder analysis, but so that is protected against extreme values, but there is loss of information or granularity. So can we do better using net treatment benefit? One suggestion can be that we use or we start with the EMA approach. So that gives you a responder if it's greater than or at least 50% seizure reduction, and then you classify the pairs, okay? But then what you can look at is also good responders, better responders, because if the reduction in seizure is 51% and if it's 90%, it's not the same, right? But they will be both classified as responder if you just look at the 50% threshold. So you can further go to a better granular level. Now, even after this, what you still have some neutral pairs remaining, because you will classify uh, the two patients who had, let's say, 12% reduction and 48% reduction, both as non-responder, but there is still that information basically gets lost as part of your analysis. And you can further integrate that by using the FDA approach, that if the change from baseline in the seizure frequency, if using a meaningful threshold of 10%, okay? So that's another way of integrating net treatment benefit in as part of the trial design. Now, by this time, you already understand that prioritizing the outcomes as well as defining these thresholds, they are two key aspects of this approach. And what we developed a tool called One to Treat Voice precisely to answer these two questions, right? So what you can do is you can select an appropriate list of outcomes using this software or this tool. You can see the order of priorities and define the thresholds as well. So this is a data-driven way that you can also specify this list of prioritized outcomes, which you can include in your design of the trial, right? We did, and I will briefly give you an explanation, and then I will demo the tool so that it becomes a bit more clear. And how this tool works is you are given two scenarios based on different outcomes and or two patient profiles, and you classify if this profile is better or that profile is better based, and then you mention like why is that particular profile is better because of which outcome. And in the end, this gives you your order of priority and the thresholds. That's what we are looking for to integrate in our design. We did that recently with OZE, and it was with site investigators, and that was in the end included in the design as an exploratory endpoint. And it was presented in ESMO last year as well. So hopefully this gives you some idea of the methodology and how the tool works. I will try to demo it now. Just before I go into the demo, so you will see pairs like this in our particular case. So that will be seizure frequency, expressive improvement, personal improvement, coping improvement, and then you have to classify which profile you prefer. Okay, so you can either scan the QR code here, or I will send a link in the chat so that if you want to try out, you can also try it out. Just give me a moment. I don't have access to the chat, Johan.
I think when you use the question and answer, Rudra, then you should be able to write something.
Okay, perfect. Okay, so I get, in case if you, I mean, I will demo it, but if some of you wants to try it as well, then please feel free to go ahead. Now, as I explained that here we consider reduction in seizure frequency, coping skill improvement that helps with the socialization of the children, then personal skill improvement, which is basically improvement in daily living skills, and also improvement in expressive skills, which is more focusing on communication, right? So you see these two profiles, and let's say I will say, okay, I prefer, as we discussed, that it's we focus more on seizures. So I will also focus on seizures, and I will say that I prefer option B. In the next case, I will still continue because this reduction is a lot, so I prefer this. I will still say 15% reduction is this, but now I see, okay, maybe there is 5% reduction is not so relevant for me. So I will look at if there is enough improvement in the expressive skills, in the daily improvement. For me, I think for me, it's more important that the daily living skills are improved. So I will say this profile is better. The same way, you get the idea, basically. I will quickly finish this. Here, if you cannot decide, you can also mention that it's a tie. So I cannot decide as well, so it will be a tie. And as you can see that as an output, it gives you the order of the priority. And that's something what essentially I had in mind, that we prioritize the seizure frequency first, and then we prioritize the daily living skills, then the communication skills as well. And then it gives you also the thresholds for these particular scenarios. Okay. So in the interest of time, I will quickly move forward because I think I have only 5 minutes more. So just to conclude that the net treatment benefit framework essentially helps us to touch all these three pillars that you see here. So it not only helps us to integrate patient preferences, or you can imagine using one fluid voice with site investigators, with clinicians, with different stakeholders, with patient advocacy groups. So depending on which stakeholder you focus on, and you can easily integrate this in the design of the trials. It also helps us to increase the statistical power or reduce the sample size in a way, and focusing on the clinical relevance. And for further reading, please, there is a book on generalized pairwise comparisons, prefaced by FDA and postfaced by EMA. So it's just to re-emphasize the fact that it's a methodology that is or it's a new approach of designing trials, but built on methodology, strong methodology as well, and where the authorities are aware of this methodology. So thank you for your attention. I will stop here. Please feel free to ask any question, or we will probably move to the panel first, Johan, and I hope I maintain the time.
Thank you, Rudra. Excellent timing indeed, and congratulations on your presentation. And I'll happily give the floor to Geert, who in the meantime was able to join. Thank you, Rudra.
Thank you very much, and nice to meet all of you. And in particular, thank you very much, Rudra, for an excellent presentation. I also took note of the book for which you made some publicity, and I think it would be a good read, at least for those people who didn't write it. But there are several around the table as well who actually have contributed to the book also, including Johan and Marc Buyse, who is on schedule for later. So indeed, what I suggest is that we move to our distinguished discussion panel. We have 4 discussants. So first there will be Martin Posch. Each of them will be introduced one at a time. Then Marc Buyse, Monica Ferrie, and Simona Giorgi. So without further ado, I would like to give the word to Martin Posch, one of our 4 distinguished panel members. Is currently professor of medical statistics at the Medical University in Vienna, Austria. So of course is connected to academia, but also to the regulatory framework. So with at least these 2 hats on, the floor is yours, Martin.
Yeah, thanks a lot for this kind introduction. And of course, all I'm saying is my personal opinion here. Next slide. So I want to focus on some of the statistical properties, but also the maybe regulatory considerations for the interpretation of net treatment benefit. So just to recall the definition, it's you can think of it sort of like an experiment where you draw a random treated patients and a random patient from the control group, and you then look at this pair. And so the net treatment benefit would be then the probability that in such a randomly chosen pair, the patient wins, so has the favorable outcome, minus the probability that the treated patient loses, so that the patient from the treatment group has the unfavorable outcome of the two. So it somehow resembles a risk difference, but of course it has a different interpretation because it's about this pairs of patients drawn from treatment and control group. Also for the interpretation, we need to have a pairwise comparison rule defined. So this is as was explained very clearly, I think, that you need on the one hand this hierarchy, so to say what are sort of the is the most important of the endpoint, and then going down this hierarchy as well as tie-breaking thresholds, so to define sort of what are ties, which outcomes are so similar that or differences are not relevant in order to define this as a win or loss. One additional important component that is needed for the definition is for time to event endpoints. So we have, for example, survival or time to progression is the time horizon, so how long the follow-up is for each patient. And in particular, if there's unequal follow-up, we need to specify how we want to handle censoring of patients, if we want to how to handle competing risks. So the interpretation of the net treatment benefit, even though maybe at first sight it seems quite simple, you just count the wins and the losses, but there goes sort of quite some definition into this endpoint that you need to take into account for interpretation. Next slide. And there's quite a lot of literature on the net treatment benefit, also discussing some statistical limitations and some oddities maybe that are connected with it. One is the intransitivity. So this comes into play if you say compare 3 treatments and you note that treatment A and B, that A is superior to B according to the net treatment benefit. So the A has a positive net treatment benefit over B. Same if you compare B to C, it also has a positive net treatment benefit, so B over C. So you would expect that also A should be better than C according to the net treatment benefit. However, that's not necessarily the case. So there are in some settings, depending on the exact distribution of the endpoints, you may end up in this situation where A seems better than B, B better than C, but C is superior to A according to the net treatment benefit. Another limitation is the lack of collapsibility, and that the net treatment benefit is not collapsible. What does this mean? It means that that the overall effect in a population is not necessarily the average of the effect in subpopulations. So assume the net treatment benefit in the overall population is 0.1, but if you divide the population in the biomarker positive and biomarker negative subgroups, you might observe at the same time that the net treatment benefit in each of these subgroups is 0.2. So this will be an example sort of where the overall net treatment benefit sort of doesn't match with what you observe if you condition, in this case, on the biomarker status. And this lack of collapsibility has been recently quite a lot discussed, not only in the context of the net treatment benefit. You can have a similar issue also with hazard ratios, risk ratios, or odds ratios, but this makes it sort of more difficult than to interpret findings in clinical trials when sort of the subgroup findings do not match the overall findings. Another issue is information loss. I mean, since we just categorize the pairs in win, tie, and losses, it means that we somehow don't care about by how much, say, the treated patient has a better outcome than the than the patient in the control group. And in addition, we count every win equally. So it doesn't matter if we win on the first endpoint in the hierarchy, which is the most important one, or the last endpoint in the hierarchy. Everything will be counted as a win. Yeah, I mean, these limitations are not just specific to the net treatment benefit. I mean, there are several related concepts or partly even equivalent concepts like the win ratio, the door, Cliff's delta, and so on, that all share sort of these same limitations. And actually, in the book that was mentioned, there's also a chapter discussing these in detail by Thomas Buschikowski. Next slide. Yeah. So what is a very favorable property of the concept is that it's very easy to perform hypothesis testing, to have strict control of time error rate. As was mentioned, it's closely connected to the Mann-Whitney-U test. So we have exact test, which is in particularly important in the context of rare diseases where we have small samples. We can also provide confidence intervals for the estimate of the treatment effect. What is clear is that we to really assess the robustness of the results, we will need sensitivity analysis. We've seen there's a lot of assumptions go in, a lot of things we have to fix to define the net treatment benefit, as the thresholds regarding the ties, how we exactly define the hierarchies, how we deal with different time horizons or follow-up times. In addition, and this is not specific again for net treatment benefit, but of course also in this setting we have to think about intercurrent events, how to deal with missing data, and how to introduce covariate adjustment. Next slide. Yeah, maybe also one point that was also mentioned regarding power and patient relevance. And we see that by aggregating more and more endpoints or more and more outcomes into a single endpoint, we have the potential on the one hand to increase power, but also to increase the patient relevance of an endpoint. However, I think maximizing power and maximizing patient relevance are often not compatible goals, or will lead to quite a different solution. Is in particular because the patient relevant outcomes may have a large variability or have low event rates. Say if survival were one of the important endpoints, but in a clinical trial, the event rates may be small, therefore the power for these most important endpoints be low. Also, if we have outcomes where the effect size goes into different directions. I mean, the example was mentioned that you can also add, for example, safety endpoints into the definition of the hierarchy. And if you assume that the safety will be worse under treatment than under control, so if the treatment also has negative effects, then obviously this will result in a negative impact on power. I mean, one possibility sort of to increase the power is to include outcomes where we expect a large effect and low variability. So, for example, biomarkers and lab values. However, they may have a weak clinical interpretability in the end, and I will come to an example where this has been an issue. Of course, this again are sort of more general considerations. It doesn't apply only to the net treatment benefit. It's a general issue in the selection of endpoints, that maximizing trial efficiency, minimizing patients needed, may conflict with choosing endpoints which have a high relevance for patients. Next slide. Yeah, I think one very nice feature of this net treatment benefit approach is this possibility to really analyze it and see sort of what contribution each of the outcomes has to the final result, and that you can really go down the hierarchy here step by step. I mean, comparing the different rows, of course, you have to be careful in interpreting sort of the change here in the cumulative net treatment benefit, because you always condition on the ties from the level before, and these are, of course, already highly selected patients and may no longer be balanced. Therefore, I think it does not replace a univariate analysis as it's sort of required in all regulatory guidance on multiple endpoints, on composite endpoints, which always also require that you look sort of at each of the outcomes separately and not only sort of in the hierarchical way. Maybe also one word of caution: the number needed to treat here is not the standard number needed to treat that you usually compare here for a risk difference, and has sort of a slightly different interpretation. Next slide. I want to give sort of one example I've found where not a net treatment benefit, but the win ratio concept was used as a primary endpoint in a clinical trial that led to an approval of medicine in the European Union. It's had the designation of an orphan indication originally. It lost it during the process, however, so because it was found that the prevalence is not low enough anymore, so it can be sort of as a kind of a borderline case. But I think the discussion you can find there is very interesting, and also shows somehow sort of the potential of the approach. The assessment report is publicly available. I put also the link on the slides. So the trial was, so the indication was a protein aggregation disorder. It was a phase 3 trial, just classical 2-arm trial, placebo-controlled. The endpoint was the win ratio, so that's not the difference between these two probabilities, but it's the ratio of the probability of winning and that the treated patient wins, or divided by the treated patient loses. And we have these 4 endpoints: all-cause mortality, cardiovascular related hospitalization, then change from baseline in a biomarker, and they also define sort of what is a meaningful change there. And then the final in the hierarchy was the 6 minutes walking distance. The result was then the win ratio here of 1.772. Next slide. And if you look into the document, they give quite some detail on the discussion of this endpoint, and their main discussion was about sort of which of these outcomes actually contributes here to the final result. And it is apparent here that the applicant and the regulatory assessors had their different opinions. So the applicant states that the primary efficacy endpoint was primarily driven by the contribution of the clinical outcomes, all-cause mortality, and cardiovascular related hospitalizations, while the regulatory assessors did not entirely follow this argument and say that the major contribution was sort of due to this lab value, this biomarker, which accounted for more than 1/3 of the wins, which was sort of seen as concerning, as mentioned before, that in this methodology, sort of each win has sort of the same weight. So the EMA then concluded that the main assessment should be based on the primary endpoint, which was actually the initially proposed one. So it seems that the company has changed it later on, which contained all-cause mortality and CV hospitalization as components only. So they agreed only on the endpoint with the first two outcomes. So it's still a win ratio approach they applied, but just on a different set of outcomes. So just the first two. And indeed, if you remove the biomarker and also then the walking distance, so the win ratio is then only 0.146. So it becomes smaller, but fortunately for the company, I mean, they also did hypothesis tests, and it's still a significant treatment effect, and the drug was still approved. So I think this is a really nice example, also showing sort of the potential of this approach, also how useful it is sort of to look then at the different levels, what each of these outcomes can actually contribute, and showed sort of how this facilitated the scrutiny also by assessors and regulators, and led then sort of to a clear interpretation and also conclusions in the end. Next slide. So before I conclude, I want to just bring up one other issue here, is regarding to the hierarchy and what to do about the challenge of heterogeneity, that if one hierarchy doesn't fit all, which is in particular in rare diseases where we often have very heterogeneous patient populations, which are maybe at very different stages of the diseases, and all are studied in a single trial, maybe of relevance. So the methods like net treatment benefit, they require that you specify a single hierarchy, which then sort of applies for all patients, so that for all patients, each of the outcomes sort of has the same order in value. But how to account for the fact that this may not be the case for all patients? So one patient may, for example, prioritize mobility as a more important outcome. Patient B, pain relief. So how to define then sort of the hierarchy when patients do not agree? In one of the earlier small populations projects in Asterix, we looked here at one potential solution, which is the goal attainment scaling, which is sort of even more radical, patient-centric, where essentially a patient-specific endpoint is defined. So each at baseline, each patient defines specific goals that apply only for this particular patient, and after treatment, when the endpoint is assessed, an independent assessor assesses then to which extent sort of these goals were actually achieved, and this then results in a score for this patient, which is then further analyzed. Next slide. So to summarize the net treatment benefit, I think it is a very powerful methodology to integrate multiple outcomes via a pre-specified comparison rule, and it comes with some complexity, and the interpretation needs to take into account the exact definition of this endpoint, including the hierarchy, ties, thresholds, time horizon, censoring handling, and also the sensitivity to these assumptions will be important in assessment. Yeah, as already mentioned, assessing the contribution of each component will be important when interpreting outcomes for the net treatment benefit, and I think the methodology also provides very nice tools to do that, and this matches very nicely also the requirements mentioned in regulatory guidelines. And finally, what mentioned on the last slide, I mean, a challenge may be if it's difficult to specify fixed hierarchies, and then maybe so which apply for all patients, and maybe these are settings sort of where each of the endpoints needs to be looked at separately, and there of course is a wide range of methods available to analyze multiple endpoints. I mean, net treatment benefit sort of is one of the approaches based on other aggregate measures based on multiple testing, and I've here cited some of the literature reviewing that. And with that, thanks a lot.
Thank you very much, Martin, for very thoughtful, insightful, and detailed comments. Food for thought as we go, but we will now go on to our second discussant, who will be a long-term colleague and trusted friend, Marc Buyse, who is the founder of IDDI, CluePoints, and 123, so well versed in industry, but of course also academically, because Marc has been a visiting professor at the Hasselt University in Belgium, related to the course on clinical trial, survival analysis for a very long time as well. So without further ado, Marc, the floor is yours.
Thank you very much, Geert, and thank you also for the speakers. As you know, I work with Rudradev on a daily basis. So we decided before the webinar that I would not present slides, but I'm very pleased to speak just after Martin Posch, who gave a very thoughtful discussion of the, the challenges and limitations and also benefits of using the net treatment benefit and or the win ratio. So I will not add from a statistical point of view, except to say that he's completely right to point to the difficulties of, for example, defining estimates. If you use the net treatment benefit, it's far more complex to define an estimate if you have several components in your analysis than for a single endpoint. And this perhaps has been underestimated by the people who have been using the win ratio for a long time. And as Martin showed, with the Beyontra example, the win ratio does have some limitations that the agencies have expressed concerns about. I think we do address a few of these, for example, the contribution of each outcome in the analysis with the net benefit. But that being said, it's unquestionable that we still have a lot of challenges with the net benefit. And so thank you, Martin, for putting these limitations in perspective. One thing that I really enjoyed in Martin's presentation is his not last slide, but the one just before last, where he said, why are we not bolder and go all the way to doing truly patient-centric analyses, where perhaps the preferences of each patient can be taken into account, in a sense, to do a patient-specific analysis. In other words, two patients might have a very different net benefit from an intervention, depending on their own preferences. So I think it opens up so many possibilities. And so this is a kind of a nice transition to the next speakers or next discussants. So I'm going to, leave a lot of time for them because they are patient representatives. And I think it's about time that we listen to what patient representatives have to tell us so that we can adapt our statistical methods to their needs. So thank you for this. And I would welcome the next discussant.
Thank you very much, Marc. Well appreciated.
Sorry, if I can take 2 minutes before Monica and Simona, just to add to something that Martin mentioned, or I can wait until the end of the panel.
Yeah, let's do it all at the end, Rudra, if that's okay. I couldn't agree more with Marc. That indeed, and this is actually also in the genome, I can say, of the RealiseD project, and runs through all of the work packages, the role of patient advocacy groups, and therefore we will have no meetings, no work without involvement of patients and their representatives. So we're delighted to have two people presenting to us now. The first one is Monica Ferrie, who is the Director of Advocacy Beyond Borders. So Monica, the floor is yours.
Thank you so much, and thank you. Can I just go back to the previous slide for a moment? Thanks. Because I wanted to say that I'm delighted to be part of this presentation and to actually have heard the presentations that have happened so far, and even the discussion. It's incredibly encouraging to hear that patient perspectives are being discussed in the way that they are. And I wanted to go back to this slide just to emphasize that I am involved at a local perspective, from a local perspective in Australia, at a regional perspective across the Asia Pacific. I'm the executive director there, and I'm a council director of Rare Disease International. I also am a director of Advocacy Beyond Borders. So like everyone in the rare disease space, and like all of you, we wear so many different hats to ensure that our message and our ability to influence is as broad and diverse as possible. So I just wanted to say, from grassroots to global and global to grassroots is the way that we all are operating. Next slide, please. Thank you. And I wanted to talk about the fact that conversation about clinical trials and conversation about measurement in clinical trials, the complexities. The patient community is really cognizant that those things exist, that this is not an easy process at all. So we do bring that into conversation as well. We know it's not simple, but clinical trials are so incredibly important to, and obviously I'm the rare disease community, so clinical trials are a gateway to a possible future for our community, and people would sell their house to get on a clinical trial, and that along the way in that investment, people die, people regress. So a clinical trial isn't happening in a vacuum where everything's traveling quite well. Life is happening as it normally would, and patients, communities, families are making a huge investment to be part of a clinical trial too. We understand that so are the people involved in funding a trial, but so is the family. And that could be in just traveling to a trial, picking up your family and moving. Certainly, we don't have a lot of trials in Australia. So Australians travel for clinical trials, which is bizarre in a country like ours. But our population, we have a population problem when it comes to clinical trials, particularly in the rare disease space. So there are lots of challenges around that. But there is also the practical challenge that being part of a trial brings anxiety and mental investment too for families, that particularly parents whose kids are part of trials, that getting them to participate in a trial on a regular basis that may involve something that child isn't particularly keen on happening can add an additional burden and pressure to a family. So that's not part of the planning that often happens is when we're talking about measures, but those things can have an impact in how families can participate. So we really want to work with clinical trial development to be able to say, well, we want the same as the trial. We want the most efficient, effective, and successful possible. And we do want an outcome, because sometimes the trials are for nothing in terms of what they mean for us at the end of the day. They obviously mean prove whether something, or provide evidence whether something works right now for everybody. But sometimes there's an investment that actually doesn't have an immediate benefit. And sometimes when a trial ends, so does access to whatever benefit you've been getting. So some of those things are the unintended consequences that go with a trial. But a trial also is part of a delivery pipeline for community, and that's how we look at it. So we want a trial, and we want a successful trial, but we also want a trial that then leads to sustainable access of whatever has been trialed. And to be sustainable, it also needs to be affordable. So we need to be thinking about, well, what is the evidence pathway that a trial is collecting, and how does that allow further down the pipeline the input of evidence that's been collected at that point? So I'll talk a little more about that. So next slide, please. So what kind of evidence are we talking about? And increasingly, we are being asked to provide, so the patient community is being asked to provide impact evidence. And that's not the evidence that says, does this make a significant difference to the condition that somebody has? It's about, well, what difference does it make to their life? How does it really matter? So when we talk about impact and want to measure impact as well, and it has been part of this discussion in, well, what are the endpoints? And the goal achievement one, I had not even heard of that being incorporated or thought about as part of trials. So that's fantastic to hear that that's part of a conversation. But because the impact for us isn't what is that drug doing to the presentation of my condition or the forward trajectory. That's such a small part. It's what actually matters is what's the benefit of that on my quality of life. So we had an example in Australia where the benefit that was the impact benefit that the patient community wanted to talk about was the fact that this particular treatment provided the ability for somebody to be able to walk 10 steps. And that was the outcome that was measured, 10 steps. But the question then about, well, what impact? How did that make a difference? The patient community said things like, it meant that I could get myself something to eat. So I could eat whenever I wanted to. And that made me self-sufficient in terms of that. I could get to the toilet by myself. I didn't need someone in my house all the time. So, and the on-flow of benefits from that autonomy and that mental health release or change in load of mental health, and then being able to think about, well, maybe I could work from home and so on and so on. So they are the benefits that we are being asked to provide evidence of in reimbursement processes. So how does that fit with a clinical trial? Can it fit with a clinical trial? That they're super important to community regardless, but we're also being asked for them. And we do understand, as I've said, and I'll say again, I'm sure, we know that it is difficult to measure these things. Next slide, please. So what do we need to get right? And yes, we know it's not easy, and that's why these conversations are so important, but it's also why we have to be deliberate and purposeful, I think, about these conversations and the way forward, because all of us have been in this space. How do we do it better for a very long time? And it's a slow, iterative process, but there are consequences to that, and for our community, those consequences are dire. So we really need to move this debate forward with some urgency about getting it right. So co-designing and partnered decision-making for what those outputs are, what the evidence interface is, a recognition that the impact, the impact isn't the process. So one of the things that we're starting to see is a vast improvement, which is terrific, in patient engagement. But that's not an end result in itself. Unless that patient engagement has an impact, unless it positively impacts, then it's not adding any value to the patient community, and it's not adding any value into the clinical trial either. So we need to measure the impact of patient engagement rather than just the fact that we're doing that, and recognizing, well, how did the fact that we work together make the trial better? Because it absolutely will make the trial better. I've talked about the importance of the evidence kind of chain, and that we need to begin that in the trial design. So what do we need to build into the trial design to down the pipeline have sustainable and equitable access and affordability? Because without those, trials and approvals really don't matter. They don't make the difference that the patient community needs them to make. It has to be accessible and affordable as well. So we also understand, and it was, as I said, that goal attainment piece was very interesting, because everyone does have different quality of life expectations. Each person may, each family may, but also culturally, geographically, there are different expectations around quality of life, and can they be part of trial design? Can that be incorporated into the way that we think about developing endpoints? But as I said, I'm so thrilled that we're part of this conversation, and look forward to moving the debate forward. Thank you.
Thank you ever so much, Monica. That's a very rich contribution. And on that note, I think we can now go to our next patient representative, Simona Giorgi, who is the scientific director at the Dravet Syndrome Foundation in Spain. Simona, it's yours.
Hi, thank you for inviting me. I think I want to echo what Monica said. I'm very happy that our voices have been included in this discussion. You can move to the next slide. Thank you. Well, these are my disclosures of conflicts of interest. While you read this, I want to make a disclaimer. I will talk about Dravet syndrome, which is what I know from the work I have been doing. I hope this can be taken as an example for other diseases. And another, unfortunately, I will talk about many problems and bring very few solutions, but I hope we can work together about this last part. Next, thank you. So just for a bit of context, Dravet syndrome is a rare disease, of course. It is caused by pathogenic variants in sodium channel, very important in the brain. And these pathogenic variants cause seizures and other comorbidities such as cognitive and psychomotor problems, behavioral issues, speech problems, and so much more. So I say this just to make you understand that Dravet syndrome is a very complex disease, and we cannot only have one outcome measure to have effective treatment. Next, thank you. The first example I bring today is the one of soticlestat, which was a drug developed by Takeda. And in January 2025, we had the news that the current data could not demonstrate the substantial evidence of effectiveness to support the approval of this drug. When we look at the data, we notice that the statistical difference between placebo and control arm was very, very little. And so we asked, and we talked with patients, we talked with their families and with healthcare professionals, and some of them, actually many of them, said that they were happy with how the treatment worked. Of course, you cannot approve a treatment based on what people say. So we looked at the outcome measures, and in the next slides, you can see the primary outcome measures that were taken into account for the trial, which is basically changing convulsive seizure frequency during the last 28 days. As I said, Dravet syndrome is a very complex disease, and the only primary outcome measures that were included were changes in seizure frequency. In the next slides, I also included the secondary outcome measures. I'm sorry about the quality of this. The yellow one are still focused on seizures, while the green one are based on CGI, on the clinical global impression of improvement, which also takes into account non-seizure symptoms, but you still can see seizures are quite prevalent. The next example, in the next slide, the next example I bring is about disease-modifying treatment. In the Dravet syndrome space, we are quite lucky. We have now 2 ongoing clinical trials that are based on disease-modifying treatments. This one is an antisense oligonucleotide called zorivinersen. When we looked at the outcome measures, the primary one is still a change in seizure frequency measured by seizure diary, which we could also talk about the accuracy of a seizure diary, but maybe this is not the right venue. But then if we look at the secondary outcome measures, there's still seizures, and then an evaluation done with the Vineland, which is a scale quite broadly used, which is not specific for Dravet, but it's the best that we have now. Here, I would like again to stress how complex Dravet is, and when we talk about disease-modifying treatments, we are talking about treatments that are changing the trajectory of the disease, mainly by changing this genetic mutation that patients present. So are we really evaluating if the disease is changing, if these are the things that we are taking into account? And I don't know, this is a question that I'm actually asking. Another example, which is in the next slide, is of another disease-modifying treatment, which is in phase 1 currently. So as you can see, the outcome measures are also quite focused on safety and on the evaluation of adverse events. And you can still see a quite strong focus on seizure frequency evaluation. And the last example is about basket trials, which we are seeing in the developmental and epileptic encephalopathy space. This is a trial developed by Longboard and Lundbeck with a drug called bexicaserin, or LP352. And what these companies are doing is one trial that includes several developmental and epileptic encephalopathies, Dravet being one of these. So the question is if one size could fit all. I think the first panelist also mentioned if we could generalize the hierarchy that we use or not. Next, please. Well, yeah, next again. So a few months ago, we actually sent a questionnaire to our families, to 50 of our families, and we asked them some questions about clinical trials design. When we asked about what parents wish to improve during as a response to a clinical trial, they said behavior and emotions, physical condition, seizures, and the cognitive area. Next. When we asked about the desired symptom improvement for positive impact on their child's life, of course, seizure reduction was important, but you can see how important were communication, attention and cognition, and autonomy, which are things that we are, in my opinion, not evaluating correctly now in clinical trials. And next, we also asked how would they consider pharmacological treatment has been beneficial, and seizure reduction was the most prevalent answer, but we also had neuropsychiatric improvement, overall improvement without side effects, and so on. And by talking to some families, I know they even consider improvement being able to go into the park, as Monica said. Sometimes just being able to do a few steps or to be able to eat could be good outcome measures. I know it's very difficult to evaluate these kind of things, but we have to talk about it and to at least try. Next. So to conclude, in the case of Dravet syndrome specifically, seizure frequency is not enough. Dravet is much more complex, and there is a real mismatch between what we are, the clinical trials we are designing, and what truly improves quality of life in the opinion of parents and patients. Next. And I actually have a lot of questions, and I would like this to open a good discussion with the attendees. For example, I know regulatory agencies now are not accepting clinical trials that do not have seizures as primary outcomes. Biomarkers have also been mentioned, but at least in Dravet syndrome, we do not have good biomarkers or good scales to evaluate other outcomes that are not seizures. Again, our seizure diary is accurate. How do we prove disease modification? How do we ensure stratification and that the needs of every patient are taken into account, and geographical equity at the same time? And I have so many more questions, but I will stop here and just leave you to the discussion. Thank you.
Thank you very much, Simona. It's a good time to thank Rudra and the 4 discussants for very insightful, deep, thorough contributions. So warm applause to all of them, virtually or in whatever way you want to give it. And I think it's now a good time, Rudra, to give the word to you so that you can react to what you heard. I've also seen that you kindly responded to a good number of questions that were posted in the Q&A, but maybe while you talk, you can also highlight a few of the conversation elements that you added there, because they may be of benefit to be highlighted again. And let's not forget about Simona's questions as well. So Rudra, please.
Thanks. Thanks everyone for the great discussion. And I think there are different aspects of overall net treatment benefit framework that's been discussed by different panelists. So first, I would like to mention that there are still a lot about this approach that I had to cut short in the interest of time. And it will be nice. So if you are interested to learn more about, feel free to just reach out. In particular, I'd like to go back to one thing that we discussed that if one size fits all, so that's it's an important question, and rightly so, because different stakeholders might have different priority orders, different it might be the threshold of clinical relevance might be different for different stakeholders, as we saw in Simona's example that yeah, seizures are important, but also cognitive scales are important. So how I see it is that we can incorporate at least to a certain extent these multiple aspects of a particular disease in the trial design, and you can have your like primary analysis based on a given pre-specified priority order. But then you can also have some sensitivity analysis to it, which takes into account different thresholds or other prioritizations among the outcomes, and that helps you to capture the full medical value of the treatment, just not focusing on the single outcome. So that's something I think is really important. And there is, so essentially you look at the net treatment benefit for different priority orders, and that gives you kind of a distribution of the net treatment benefit. But I will stop here to also see if Marc wants to add something.
Not at this stage. Thank you. I think you've covered a lot of grounds.
No problem. Thanks, Marc. So that's one. Then I tried to answer some of the questions that's answered in the chat. I mean, that was asked in the chat. And as purpose of the demo, I showed a very simple way of the tool that essentially gives you the opportunity to discuss with key opinion leaders or experts in the field to capture inputs from them. And in the demo, I just fixed it on for specific outcomes, but it's very flexible, and we can customize it to select or identify outcomes based on the inputs from different stakeholders. So you can have a list of outcomes and you ask them to select certain outcomes from the list, and in the end it tries to the algorithm behind the tool tries to identify the most representative hierarchical ordering, let's say. This is just to give you a bit more explanation about the tool. And again, in this context, feel free to reach out if you want to discuss the tool, or if you want more details about the algorithm behind, right? And we try to the design of the tool in such a way to represent the patient profiles as realistically as possible. So for example, that you cannot have a profile where you have serious adverse events, but your quality of life improved, right? So there are certain constraints behind it, and we try to keep this in mind. So the conclusions that you draw from this inputs from different stakeholders using this tool, we can really use this information to pre-specify as part of the design, to look at different aspects of the trial. We have 2 more minutes. I will stop. And if you want me to focus on any specific question, I'm happy to answer.
Thank you very much. Let me at this stage actually abuse my position to also make a few very brief comments, also provoked by Simona's very important questions. For example, the one about EMA, FDA, of course, I'm not going to answer it. I cannot, I'm an academic. But I think RealiseD in a way is the answer, because EMA is a partner in the project. So that means that in the various work packages, including ours, work package 5 on statistical analysis methodology, work package 4 led by Dieter, who is also present, more on design aspects. All of these are co-created with both patients and regulators, of course, next to academia and industry. So I think in that conversation, these questions should be center stage all the time. And then, of course, we can basically move towards an answer together. Everybody plays their role, but I think we have a marketplace for interaction that we haven't had before. And of course, I'm not talking about the final answer and the solution, but rather the road leading to it. I think I'm not an expert on Dravet syndrome, but I had the opportunity with other people also to work a little bit on it in the past. Are seizure diaries accurate? I think asking the question is kind of answering it. And of course, it points to the fact that we need combined evidence, but that in a way was the topic of today's presentation. And there are also other methods indeed as well. With that, I think I have the pleasurable task to conclude a very fruitful webinar, I think, with excellent discussion contributions following the presentation by Rudra. And on that note, I give the word back to the overall chair, Johan Verbeek.
Thank you, Geert, and thank you everyone for listening. I hope it was very useful for you. And of course, I want to thank the speaker, Rudra, all our panelists, Martin, Marc, Monica, Simona, under the good guidance of Geert. I think we heard a lot of good points, but also a lot of room for co-creation to move forward with this area. So I think that is very hopeful for RealiseD. And I thank you all. Wish you a very pleasant rest of the day, wherever you are. Thank you very much.
Thanks, Johan. Bye, everyone.
Bye. Thank you.
You.